Hello everyone. Hello ma'am. I am here, Annu Yadav. I am Annu Yadav from the group number 316. I am here to discuss about some topics like electrolytes, composition, and the primary structure of the protein, and the nucleic acids. So we will first go through the primary structure of the amino acids, proteins. So here are the first topic. Now, we know that proteins are made up of amino acids, and amino acids are bound together by the peptide bond to form the protein. Okay, the molecules that are less than 100 residues are usually called as the polypeptides rather than the proteins. Okay, and we see that the protein has primary structure. The protein has two terminals, like N terminal and the C terminal. N terminal is formed by the amino group, and the C terminal is formed by the carboxylic group. Okay, the N bond is partially the NC bond is the partially the double bond, and CO bond is partially the single bond because of the electron density distribution around the N and the O molecule atom. Okay, we see that when we eat food, there is a hydrolysis of the protein occurs by the enzyme present in our stomach that we call proteases. uh, in this uh, in this digestion, we have many proteolytic enzymes, and these amino acids are mm, mm, we get the solution of the amino acid, we can say that, and it is analyzed by the chromatographic techniques. So we got the many amino acids like tryptophan, methionine, histamine, alanine, serine, glycine, valine, and many so on. For example, trypsin hydrolyzes only those peptide bonds which are formed by the CO groups of the basic amino acids. Example of the do you remember the example of the amino acids, basic amino acids that are arginine and lysine? So we get the mixture of the peptides through short fragments of the peptide protein chains. So we will see the next topic, that is I have written here the protein text reflects the primary structure of the protein. Okay, so the primary structure of protein is kind of a text written by the 20 letter alphabet. Anyone can guess what is this? So I will explain. So the sense and the content of this text I written here is normally refers to the biological function of the protein, which is determined by the final analysis of by the primary structure. The protein text reflects the biological evolution. That means the homologous proteins which perform the same function in the different species. Um, and we can see the evolution through these type of examples, like cytochrome C is in the respiratory chain is present in the all organisms. Okay, we will see that the primary structure of the protein can also vary as a result of mutation, and there appears errors in the protein text which have as the rule, like for example I have written the sickle cell anemia is an inherited disease is caused by the substitution of the valine for glutamic acid at the sixth position of the beta chain hemoglobin. The man's hemoglobin is made up of the four chains. Two are identical alpha chains and two are identical mm, beta chains. I have written here the alpha chains contains 141 amino acid units, and the beta chain contains 146 amino acid units. In this, the non-polar neutral acids, that is valine, is replaced by the acidic units. So this hemoglobin is get mutated, and it is kind of like dysfunctional. Okay, so primary structures are the baseline of the protein formation. So we can go ahead and we will see the next topic. This is nucleic acid. Anybody know what is nucleic acid? We already studied in our semesters, in earlier semester. Nucleic acids are the baseline of the DNA. Nucleic acid forms the DNA. Nucleic acid can be can form RNA, DNA, whatever. Okay, the nucleic acid, it's formed by three units. That is, first is the phosphoric acid, second is the sugar, third is the nitrogen base. Sugar can be of the many forms, like carbohydrate ribose in the ribonucleic acid, deoxyribose in the deoxyribonucleic acid. Deoxy is the oxygen is not present in the DNA. We know that deoxyribose, this when this oxygen is not present, here we can see that this oxygen is present at the second carbon of the ribose sugar, and here is not any oxygen is present. So it is a type of inert. It is non-reactive, and this ribose sugar is reactive. It is mostly present in the RNA, and this is mostly present in the DNA. So phosphoric acid unit, sugar and base.